Welcome to video 2.5. In this video, I'll be covering the stability and the differences between DNA and RNA polymers, now that we understand structurally what they look like and what they're made of. Importantly, the 2'OH will be front of center here as a big difference between the two. But before we dig into the specifics, I want to focus back on something I brought up in section 1 of the course. One of the key ideas I like to focus on when I'm teaching students about biochemistry is the importance about how structure dictates function, and that if you want to understand a structure, it can be very helpful to know what the function is first, right? The structure is not randomly evolved. The structure is made and evolved to have a specific function. So the question is, what are the different functions of DNA and RNA polymers, and how do their structures cause this difference in function, right? Because all functions are caused by differences in structure at the fundamental level. So let's do a quick review to answer that question of, intro biology, right? DNA is found in the nucleus of the cell. So keep it blue. Here's my DNA, right? And what is its function, right? It functions as a store of information, right? We learned from the Hershey-Chase experiment, the initial proof of that. But the important idea is that we need this DNA, right? It's going to be with us our whole life and constantly being read like a blueprint. And we're going to be using that to, develop from a embryo to a baby, from a baby to an adult, and for an adult to continue living. The DNA needs to continually store and be read for information, right? That's where DNA is. Now, RNA is found in the cytoplasm, and nucleus. But the goal of RNA, right? RNA is copied. We make copies of RNA from DNA, and then we export those copies into the cytoplasm. The mRNA is then read by ribosomes, and proteins are produced. Right? So DNA encodes RNA. This is the central dogma of biology or biochemistry. DNA encodes RNA. RNA makes proteins. Right? So the function of RNA is not to store information, really. It's to, move information, to transfer, to message information from the DNA to the ribosomes, right? So that's the key difference in their function, right? And why does that matter? Well, the key idea here is that DNA must be stable. Oops, there we go, stable. It must be stable to be a useful store of info. If you're going to store information, and we want that information to be there with us our whole lives, hopefully 80 plus years, right? Mutations in that DNA sequence, if it's unstable and it starts changing, that could change genes that are incredibly important for survival. That could kill you, right? Or cause cancer or all sorts of things, right? So we want our DNA, in order to be a useful store of information, right? That's its function. It needs to be stable. Meanwhile, RNA, if we think about it, we want it to be unstable. And that might seem interesting, right? Why would we want RNA to be unstable? Well, remember that its role is to be a messenger of information. Let's say that you run across a tiger. This is, there's a tiger. And this is this is an analogy. This isn't how biology works for this. But that response, there's a bunch of, nerves and triggers and all these crazy things that stimulate some sort of change in gene expression, right? We need to run. So we need to make a bunch of adrenaline. We need to, all these things happen. So exposing ourselves to the tiger, right? That's going to lead to a change in the RNA, the DNA that's active, that's producing RNA. And so maybe this piece of RNA here, right, is produced, and its job, right, the protein that it encodes for, is to produce adrenaline, right, to make you run really fast and not feel any pain and escape the tiger. Let's just pretend that's the case here, right? Again, overly simplified. Well, then the tiger goes away. You've ran away from the tiger. Great. The issue is, we still want to have that template DNA, right? The DNA that can help us if we run into the tiger again in the future. We're going to want that present in the nucleus. The issue is that this RNA, right, that's actively being translated into protein. we don't want to keep making adrenaline and keep reacting as if the tiger is there if the tiger isn't there anymore. So we want this RNA to be constantly degrading, right? If I want a protein to be consistently made, what I should do is I should continue to make more RNA. And as it degrades, more is made. And as this one degrades, right, more RNA is made, right? That translates to the proteins, and we can control the genes, right? So we actually don't want our RNA to be stable, because then it would keep functioning for way too long, right? It's supposed to message information and then be destroyed. So hopefully that makes a little bit more sense as to the function of these things. DNA must be stable and RNA must be unstable. So how does RNA's structure make it unstable is the next question, right? The central line here is both DNA and RNA can be hydrolyzed, right? They can be broken down with water. So if you apply enough water and enough base or acid or something, you can break down DNA. But RNA can self-hydrolyze. And this is really important because it has a 2' OH. And remember the key idea, right? Structure dictates function. The reason why RNA is going to have that extra 2' OH, right? So if we look back here, right, here's RNA. It has that 2' OH on carbon 1, 2, where DNA does not, right? Up until now, that's how we've recognized RNA and DNA, but why is it there? Right? The answer is that this OH makes this molecule much more unstable. RNA is unstable, DNA is stable. So let's break this down in a little more detail. Hydrolyzed means broken by water. So DNA and RNA, if exposed to water and the right conditions, in theory, the water can break the phosphate bond, and you'd basically separate this DNA base from this DNA base, and you'd break your chain. You'd lose your information. There are enzymes called DNases, or DNA aces, enzymes that break down DNA that make this a lot easier, right? Meanwhile, for RNA, the same thing can happen, right? We still have the phosphodiester bond. It can still be broken by water. So for this reason, right, again, the idea that both DNA and RNA can be hydrolyzed, but RNA can self-hydrolyze. Let's break that down in more detail. So here is a piece of RNA. We have our two sugars, A and U because there's RNA, and we see that there's OHs. Now, one of these OHs, the 2' OH up here, I've made it negative just to make this make more sense chemically, but regardless, this oxygen is in position to directly attack the phosphate. So don't worry too much about being able to remember this exact set of arrows and pathways, but I do want to show the organic chemistry behind this. The oxygen can attack the phosphate, and what that'll do is it'll lead to the formation of this new ring. So you'll see this new ring here, right? The idea is that this oxygen is now also connected to the phosphorus, right? DNA could never do this because DNA doesn't have this OH. So this is only something that RNA can do. Now, this arrow pushed some electrons up to this oxygen, so they'll go back down and we'll kick out the bottom sugar. What that leaves us with is two distinct chemicals, and we've now broken our DNA, or the RNA rather, has been broken apart. On the bottom, we have an RNA, so this is our, uridine sugar, right? We have our, whatever was on the 3' end, you'll notice, gets cut off. And on the 5' end, right, the part that was closer to the phosphate, we now have this cyclic phosphate, which is kind of what I like to call it, right? We have our sugar with our adenosine. We have our, phosphate group. But down here, we have this weird circular ring we've made with the phosphate. So that's weird. At this point, by the way, the RNA is already broken down. But we can even go further. That cyclic, so the 2,3, so carbons 1, 2, and 3, the 2,3 cyclic nucleotide monophosphate, I guess it's a diphosphate in a sense since there's two of them. But what happens here, right? You can produce one of two chemicals, right? Either the phosphate ends up on carbon 2, or the phosphate ends up on carbon 3. So you can get a 2-phosphate or a 3-phosphate. Either way, the RNA chain that you made has been broken down into individual things, right? RNA just does this by itself. So again, the key idea here is that DNA is meant to be stable, RNA is meant to be unstable. The reason why RNA has that OH group there is to help it become unstable. It can react by itself to destroy itself, to ruin. If I have a string of information, that can break it. And an interesting note here, just a quick note before I wrap to the next topic, is on mRNA vaccines, right? COVID vaccines, we've heard about mRNA technologies. The basic idea is that you have some sort of lipid particle that's able to fuse with your own cells, and inside it has a piece of mRNA. So when this goes into your cell, right, here's your cell, and the mRNA is present from the vaccine, your ribosomes will just translate that into protein. Right? That protein is the protein that your body will recognize and attack, and that gives you immunity to the COVID virus, or not total immunity, but good outcomes. It's a good vaccine. So with that in mind, right, some people have said in the news, and people that are less scientifically educated, that mRNA might be in your body forever. But keep in mind that mRNA vaccines, by the nature of the chemical, are self-degrading. So they'll be present for a while. They'll be there for the vaccine effect, but then they will self-degrade. And that's kind of one of the cool things about these vaccines is, they're going to degrade. And I wanted to bring that up as part of this topic. So without further ado, we're almost done the nucleic acid section. We just need to cover a few more things about the secondary structures of acids and some techniques and research for them. But hopefully, at this point, you feel a little stronger about your nucleic acid background and more equipped to handle questions on your upcoming test. Anyway, that is if you're taking a test. If you're just curious, I'm glad you're learning. I'll see you in the next video to discuss secondary structures of nucleic acids.